and welcome to Unit C, Lesson 12.2, Inheritance and Genetics. So when we think of inheritance and genetics, we often think of modern day stuff because there's so much that we know now about DNA, and we can even go in and tweak the DNA of like plants so that maybe a crop that we're growing for wheat has characteristics that allow it to grow faster in the colder climate of Alberta, something like that. But genetics in plants especially goes much further back than that. And so we have to really start by looking at the work of this fellow, Gregor Mendel. And so he's basically watching some pea plants that he's got growing, and he notices that consistently some of them grow tall and some of them grow short. So he figures there's something encoded in these plants that tells them whether they're going to grow tall or short. So he starts keeping really careful records about all of these plants. And we can do something with plants that is just like with humans. You can have plants where you cross fertilize them with each other, and you basically have the two plants you started with as parents, and the plants that they create from like seeds that you gather or whatever are the offspring. So that's exactly what Mendel does. So what he does is he starts trying to take tall plants and crossing them with each other as parents. And what he finds is that in some situations, some of those tall parents always related or resulted in tall offspring. So he figures that those tall parents, since all of their offspring plants are always tall themselves, they must have only the encoding, the information of how to grow tall. That means that the parents that he started with are purebreds. They only have a set of information for how to grow tall. Then he tries the same sort of thing with a bunch of short plants, and he finds again that there are some short plants that when he crosses them with each other, he always gets short from their offspring. So that must mean that those short plants were again purebred. They only had the information for shortness. So you could have purebred tall plants or purebred small plants. The way that we would look at it today is that the purebred tall have only genes for growing the specific trait of being tall, and those purebred short have only the specific genes for making the short trait. So basically, Mendel asks himself, what happens if we start trying to cross some of these plants and see what else happens? So he takes tall parent with a purebred short parent and crosses them. And what he finds is all of the offspring are still always tall. Now he knows somewhere in there is the information for how to grow short plants, but none of the offspring ever showed shortness as a trait. So what he figures is that sometimes certain traits are what we will refer to as dominant. In this case, tallness is a dominant trait. Even though those offspring were getting tall and short genes, only the tallness ever showed because the dominant gene for tallness always wins out. It overrides the recessive trait of shortness. Even though that genetic trait is there, the genes for it are there, because shortness was a recessive trait in these pea plants, it just wouldn't show up if there was tallness there. Now, those plants, because they're carrying tall and short genes, even though we only see the tallness, we refer to them as hybrids because they still carry information for both tall and short. So Mendel keeps, playing around with this stuff, and he starts taking hybrids and crossing them with each other. He notices that most of the plants still come out as tall, every so often there would be a short plant. That's his proof that there must still be the information for short genes in there. It's just that they're not coming out very often. But he kind of wonders, why do the hybrids of all of these plants allow mostly tall to come out, but every so often a short plant? That means that what we have to do is start to keep track of all of this genetic information that these plants are sharing with each other. We're going to create something called a genotype. And when we do a genotype, what we're doing is assigning letters to certain traits so we can keep track of the genes that are making these traits appear. Now, the rule is if we have a dominant trait, like the tallness that's happening, we're going to give that a capital letter for its gene. So in this case, we're going to use capital T for tallness because that's the dominant gene, that they're going to be tall. We're going to assign a small letter to the recessive gene. Now you might say, are we going to use S for short and make it a small s? No, we actually use the same letter as we did for the dominant gene. So I know it might seem kind of weird, but we're going to use a little t, and that's going to be the recessive gene that represents shortness. Capital T for tall, short, or small t, I should say, for short. Now, during reproduction of plants or animals, the genes that we get from our parents, we get one from each. Each parent gives one or the other of its genes. So what we have to do is now have a way to kind of keep track of all of this so we can figure out, like for example, in the hybrids, why we got still mostly talls, but every so often a short. That means we're going to have to draw out what we call Punnett squares. This is something that a guy named Reginald Punnett came up with. And what he said is we're going to kind of play almost like a game of Battleship. Along a top row horizontally, we're going to list the traits of one of the parents, the genes it has. Vertically, we're going to show what the traits are, the genes are from the other parent. And then what we can do is we can see how the parents give the genes to the offspring, and we get the possible combinations of what can happen. So this is a Punnett square. Now please understand that if we get into like really high-end genetics, they're obviously going to be, potentially more complicated than this. But this actually works just fine when we're trying to keep track of simple traits like tallness and shortness coming from parents. So up along the top here, we have one of the parents, and it is a purebred tall, so both of its genes are tall. On the other part, on the sort of the vertical part, we have our other parent, and again, we have a parent that is purebred tall. Now, what we need to look at is how we get combinations of offspring in these boxes down here. So in this box right here, I'm looking at how I get this gene from one parent and this gene from the other parent. So in this box, I get tall. That means that that offspring, because it has both tall dominant genes, it's going to be tall. In this box, I have tall from this parent and tall from this parent. I hope you can see how I'm saying it kind of looks like I'm playing Battleship. So I again have tall for that offspring. It will be a tall plant. Down here, I've got tall from this parent, tall from this parent. I've got tall again. It's going to be a tall plant. And I think you can see where this is going. Final box, tall and tall. In this box, tall. I've got a plant that will be tall. So if I have two purebred tall plants, I get all tall offspring, and they themselves are purebreds. All they can ever do is pass on to other offspring two tall genes. Well, one from each of the parents at least. Now, what about if I start with both parents being purebred short? So I've got one parent up here, and it's got small t for short, and small t purebred short over here. In this box, I'm getting short and short. In this box, I'm getting short and short. In this box, I'm getting short and short. See where this is going still also, right? And short and short. Yeah. So if I have two purebred short plants, I'm going to get a whole bunch of offspring that only have the short gene, so they're all going to be short. Okay, so this is just like Mendel was seeing when he did his experiments. So what about if I take a purebred tall and a purebred short? This actually did come up with a different combination that we discussed. I'm getting tall from this parent, short from this parent, so that's going to be tall short. In this box, tall from this parent, short from this parent, tall short. In this box, tall short. And just so you know, we always write the dominant gene first and then the recessive gene. And then in this box, tall short, tall short. So this is the combination that we called hybrids. We have purebred tall with purebred short, giving rise to a hybrid set of offspring. All four of these offspring carry both the tall and the short gene. But because tall is a dominant gene, all of these offspring still grow tall. They have that almost like hidden recessive gene that could allow shortness, even though it's being dominated right now by the dominant tall gene. Now we're going to do the last combination, hybrids. We've got a tall short parent up here and a tall short parent over here. So in this box, we've got tall tall. That plant is going to grow tall. Over here, I've got tall short. Because tall is dominant, it will be a tall plant. In this box over here, tall short, and I've got another plant that has the dominant tall gene, so it will be tall. But here, this is the special one, because it's getting two short genes. Each parent gave a short recessive gene. This plant will be short. Remember when we talked about the hybrids, we said that we got a lot of plants that were tall, but every so often a short plant would sneak in. So what this basically says is we're going to have a purebred tall, two hybrid still tall, and one purebred short. Now this might sound like it's going to come out this way exactly every time, you know, four plants with exactly one of them being short. Now, this is all based on probability. It's like flipping a coin. If you start flipping a coin and you do it only like 3 times, you expect to get, you know, a couple maybe heads, one tail. You could, though, get like 3 heads in a row. But if you start flipping a coin like 300 times, you expect to get about half of them to be heads, half of them to be tails, even if it's not exact. And that's the probability we're seeing here. We basically have a 1 in 4 chance of getting a purebred tall, and it's going to be tall. We have a 2 in 4 chance of getting a hybrid, which will still look tall, it'll still grow tall. And we have a 1 in 4 chance of a short plant. And it'll actually grow short because it only has those short genes. This is still all probabilities, though. It doesn't mean anything is certain, but it means those are the possibilities. Now, we can look at this as an example of how genes can actually be transferred in humans. There's a condition known where a person can be albino. An albino person has recessive genes that mean that they're not producing a lot of the pigments that other people will produce in their skin, in their eyes, in their hair. So typically somebody who is albino will have pale skin. Their eyes might have um, color to them that seems not quite exactly the same as other people have. They might have a reddish tinge to them, and hair that might be blonde, very whitish, in fact. Now, this can be because the parents, even though neither of the parents is albino, they were carrying a recessive gene that allows for someone to be albino. It might be that since both of them are carrying that recessive gene, they could still have like a 3 out of 4 chance that their child would be born without being albino. There's a 1 in 4 chance that the child could be born albino. And that means that what parents can do in our modern day world is actually go for genetic testing, and they can see if they're carrying any recessive genes. Now, sometimes these recessive genes can actually be related to very serious, life-threatening, genetically transferred diseases. Then what the parents have to do is come to some sort of a decision about whether or not they want to have children, when they have to consider the chance of their recessive genes being passed on and potentially giving a life-threatening disease to a child. Now, what can also happen out of this then is something called pedigrees, where a geneticist, a person who specializes in genetic traits and dominant recessive genes and all that sort of stuff, can look for patterns in the genes that are being passed on. This can mean that they look back many generations, um, you know, parents, grandparents, great grandparents, to find out if there were any particular characteristics that were being shown that might be associated with a recessive gene. Or is it all like purebred genes or dominant genes, I should say? Pedigrees are something that are actually quite important when you're talking about buying dogs sometimes. So you might hear that a dog has its papers, and that means that what they've done is tracked that dog's parents, grandparents, great grandparents, going back several generations. And so what they can say is that, for example, you're buying a bulldog that is a purebred bulldog because we can trace back many generations where all of the parents, grandparents, great grandparents were themselves all bulldogs. And that nowhere along the way did like a Chihuahua be one of the grandparents or something like that, because that would mean that there is genetic information in the dog making it not a purebred. And for some people, this is really important as far as having these dogs that can be shown to have, very particular genetic traits that have been carried for many, many generations. That's all that we're going to be looking at with this. Hope you have a good day.